Hello and welcome to another exciting lecture for Foundations of Behavioral Neuroscience. In this lecture, we are going to be finishing up Chapter 2 on the structure and functions of cells of the nervous system. This should be a relatively short chapter, but as I said when we did the first part of this chapter, that I like to break it up just because of how much information I'm throwing at you. So looking at what we're going to do in this chapter, we're going to look at communication between neurons. We're going to cover synapses and the synaptic gap and the presynaptic and postsynaptic and all of that fun stuff. We're going to do a brief introduction to neurotransmitters, yet unlike what I asked for in the discussion this week, we're not going to get into neurotransmitters that much because we will get into those in detail in Chapter 4. Then we're going to talk about postsynaptic potentials, and finally finish up a little bit with deactivation. So you should recognize this picture from the first half, and basically this picture is referring to different things like the sensory neuron, the interneuron, the motor neuron, and how all of those work. But we're actually going to look at this a little bit more in depth. So what is actually occurring when this system is working? When it's just going from the sensory neuron to the spinal cord, through the interneuron to the motor neuron. What is happening here is that a reflex action is occurring. It skips the brain, and we'll come back to this later, but it skips the brain and a reflex occurs. So you touch something hot and you pull away from it before your brain even registers that you've touched something hot. It's very important evolutionarily that in certain circumstances that you act before you have time to think. However, one of the issues here is that this causes problems in that sometimes you act in ways that are counter to what would be appropriate or what you would want to do. And we're going to look at that and how actually the brain can modulate that. So with this simple neural circuit, you can actually have inhibition. Inhibition is where the brain can override reflexes. So when you touch something hot, if you're not thinking about it, if you're not aware of what you're doing, that inhibition, it's really tough to work with. Inhibition is an active role, so you have to be actively inhibiting before even the stimulus that causes the reflex. So you have the stimulus that causes the reflex, normally you reflex from it, you pull your hand away. But if you are actively inhibiting, so inhibitory signals arise from within the brain and can prevent this withdrawal reflex from causing a person to, in this case, drop a casserole. When the brain knows that you're about to touch something hot, you override this reflex. And that is, if you can see it in here, where it's right in there, the interneuron, this interneuron, it's a different interneuron. It's an interneuron that's basically getting activation from the brain, is actually acting to inhibit this reflex action. So the muscle, the sensory says heat, pain, ow. The interneuron relays that to the muscle and the muscle pulls away unless there's something else there that is inhibiting that muscle reflex. And when we look at the neural circuits and how things like that, you'll start to see what is going on with inhibitory signals. So let's look at now the communication between neurons. And there's some important things going on here, but it's actually relatively simple. That is, synaptic transmission is a means of communication between neurons. you have the synaptic gap. You have this synapse. So the synapse or the synaptic gap is the gap between between basically two neurons. Whenever there's a neurotransmitter working, whenever there's something that's basically the presynaptic cell is working on a postsynaptic cell, you have a gap between them, and this is called the synaptic gap. It's a fluid-filled gap which chemicals move through. And these various different chemicals, which we're going to talk about later, are neurotransmitters. And we'll even get more complex later and talk about how some things that aren't neurotransmitters act as neurotransmitters. Some neurotransmitters can actually act in other ways than just as neurotransmitters. But when we're talking about the synaptic gap, we're talking about neurotransmitter molecules. And these neurotransmitter molecules go to the postsynaptic cell and cause postsynaptic potentials. So in the previous lecture, we talked about how you get depolarizations and hyperpolarizations of the neuron, basically causing it to fire and then reset. These neurotransmitters actually aid in that. They do more than that, but the basics of it is these neurotransmitters are typically going to cause either a depolarization or a hyperpolarization. They are going to either cause a cell to fire, stop a cell from firing, or bring a cell back to normal after it's fired. Those are the three main things that are going to happen. Okay, I skipped ahead accidentally when I double-clicked there. It's really difficult for me to go back when I'm recording in PowerPoint because I have to re-record the whole slide. So I'm just going to say what was the rest of that last slide, and you can go look in it in your slides. But basically, what neurotransmitters do is they exert effects by attaching to binding sites. Those effects that I was just talking about, they exert by attaching to binding sites on the postsynaptic cell. And they fit together like a lock and key. So the picture that was on the previous one showed a whole bunch of keys as neurotransmitters. Neurotransmitters aren't shaped like keys, but neurotransmitters do have specific shapes. Each one has a different shape. And the sites, the binding sites, are each shaped in certain ways so that neurotransmitters of certain shapes will bind to them. So neurotransmitters of a specific type will go to specific binding sites, and specific binding sites are shaped like that. So they fit together like a lock and key. This is actually something we'll come back to talk about later, how you can actually have other chemicals that bind to these sites but don't fit perfectly, so they don't cause the binding site to activate. But what they really do, what their main point is they block neurotransmitters from binding there. And it basically shuts down that neurotransmitter in a lot of ways because they're blocking the neurotransmitters from working. And finally, the last thing on the last side is ligands. Basically, ligands are any chemical, including neurotransmitters, that bind to these binding sites. Okay, moving on now. Sorry about that, but it ended up double clicking. So now let's talk about this slide. On this slide, we're going to look at how these synapses are connections between neurons, as we've been saying. They're the space between terminal buttons on one neuron and the dendritic or somatic membrane of another. However, there's other times and places that they'll be. This is the typical. So what you see here, what it says here, where you've got between terminal buttons and the dendrites or the somatic membrane of another neuron is this is what's typical. But this isn't actually always what happens. And we'll look at some of those later. You do have dendritic spines, which are basically spines that come off from the dendrite that basically are where terminals can connect to. It doesn't have to connect to a dendritic spine, but that is one place where they commonly connect. In the presynaptic membrane, it is what is coming off from basically the terminal buds of the presynaptic neuron. The postsynaptic membrane is the neuron that's receiving, or the neuron that is after the synaptic gap. They face each other across the synaptic gap. And then another important thing here is, and there isn't a drawing of it here, but you have what are called synaptic vesicles, or vesicles. Yes, I can speak correctly today. Synaptic vesicles. And these are made of a membrane and filled with molecules, usually neurotransmitters. These are in the terminal buttons, so they'll be in the terminal buttons, and they'll just be all over in this terminal button, even all the way back. You can have hundreds of them, even up to millions of them. There's some that are closer to the surface. There's some that are right on the surface but aren't open. And basically, when these make it to the surface of the terminal button, they release the chemicals, the molecules that are in it, the neurotransmitters that are in it, into the synaptic gap. Actually, on the next slide, I'll show you a zoomed in picture of that. Now, we're not going to go into these, but you've got axodendritic, axodendritic, axosomatic, and axoaxonic synapses. These are basically different types of synapses. I'm not going to test you on that specifically, but the axodendritic are ones that go from terminal buds to dendrites. The axosomatic go to the somatic membrane of the receiving neuron, and the axoaxonic go to the axon of it. Or they can even go to other terminal buttons. So what you see over here in D is actually one of those exceptions to that, when I said that it usually goes from the terminal button to the dendritic or somatic membranes. And in this case with D, it's one terminal button on top of or going into another terminal button. So the synaptic gap is between the two terminal buttons. And we'll talk about this later and what this actually does to the terminal button that it's connected to. So the postsynaptic terminal button and how the presynaptic terminal button affects that. I said we'd have a zoom in on that. So let's look at these vesicles. Synaptic vesicles are all of these in here. You see some are close, some are just opening up, some are actually open and have released their chemicals. They release it into the synaptic cleft or the synaptic gap that you see here that's down inside. That synaptic gap is again where these are released, and it is where the neurotransmitters go through to the postsynaptic membrane or the postsynaptic terminal. We're going to, in just a little bit, look at what goes on with that postsynaptic terminal, but it's by the process of diffusion, these neurotransmitters that are being released in high concentration on the presynaptic terminal are moving through the synaptic gap to the postsynaptic terminal. Okay, so like I said, we're going to talk briefly about neurotransmitters here, but in Chapter 4, we're going to talk about them in much more detail. So neurotransmitters are basically a chemical that's a messenger that is that key-shaped. They're different shape, they're that key-shaped. They're released by the synaptic vesicles on the presynaptic neuron. They go to the postsynaptic neuron and bind to sites that are specific to them. So they're released when the synaptic vesicles fuse with the membrane and break open, spilling the contents into the gap. And through the process of diffusion, which we talked about in the first chapter, in the first half of this chapter, they move across the gap. Then you get receptor activation. This is where these neurotransmitters diffuse across and attach to specific binding sites called postsynaptic receptors. These postsynaptic receptors, again, are specific to individual types of neurotransmitters. So you'll have one that's for one neurotransmitter, but you'll have other ones nearby that are for another neurotransmitter, and other ones that are for another. And these different neurotransmitters are going to have different effects, because once that binding occurs, the postsynaptic receptors then open neurotransmitter dependent ion channels. So if you'll recall back to the chapter or the lecture from earlier this week, ion channels are opened up in the neuron to basically facilitate the firing of the neuron. But these ion channels, basically what they do, which ones open up, is going to affect what occurs with that neuron. If the neuron hasn't fired yet, and you have sodium neurons or sodium ions that are allowed through, so the sodium ion channels are opened up, then that's going to likely cause the neuron to fire. However, what happens if potassium ion channels are opened up? This is actually going to make it so that the neuron is less likely to fire. And there's actually other things to consider here. There's chloride. There's basically multiple different ion channels, more than we even covered, that can open, that can basically do various different things to the neuron, basically increase its firing, decrease its firing, and some of them will even some of these ions go through, do less to increase or decrease its firing, and actually do other things like go into the receiving neuron and cause that neuron to change in ways, cause it to maybe create more neurotransmitters, because if you recall, the soma of the neuron creates neurotransmitters. So it's just, there's a lot going on here, a lot even beyond what we're talking about. So the movement of these ions changes the local membrane potential, possibly causing the neuron to fire, and there are direct and indirect methods. Let's get to the direct first. So direct ionotropic receptors. These are ones where your neurotransmitter here binds to a direct receptor. It binds to a direct postsynaptic receptor. These are really easy to explain, and they're really easy to think about. It binds to that, and it opens up the ion channel. Very easy, very simple. It goes there, it opens the ion channel. If there isn't one there, like here, the ion channel is closed. If there is one, the ion channel is open. So it's very easy, very simple to think about. If a neurotransmitter that it will open up a specific ion channel binds to a direct receptor, a direct postsynaptic receptor that opens that ion channel, it will just open that ion channel up. It takes very little energy from the neuron for this to occur, because it's almost a mechanical thing. It's almost like pushing a lever. That's direct. That's the easy one. The indirect, or the metabotropic receptors, are much more complex. There's two different types of these, and that is, in the first two steps are the same. That is, the molecule binds with a receptor. This receptor does not directly activate the ion channel. Instead, this receptor activates a what's called a G protein. It activates a protein inside the neuron. This protein then goes and binds inside the neuron to, in the first case, inside the neuron, to a ion channel, opening it up in a mechanical way, just like if it was binded to the outside. We'll talk in a minute about why this is more complex, and what occurs, and the difference here in why this is occurring. But it comes down to, like I said, we'll talk in just a minute about what's going on and why this more complex method is here. But just note that this indirect is more complex. So it hits the receptor. It causes the receptor to activate a G protein. The G protein is then released and hits the postsynaptic ion channel, but it hits it inside the neuron, and it opens up that ion channel that way. This also requires less energy, well, not as much energy as it could, but it does require more energy than the direct. That is because this part here requires energy to work, even if this part here doesn't. All right, let's look at the other one that's even more complex. That is, we get the neurotransmitter binds to a receptor. The receptor activates the G protein. The G protein breaks away and then activates an enzyme. It doesn't go to to the ion channel. It goes and activates an enzyme. So it basically binds to a site that activates an enzyme. This enzyme produces a secondary messenger. Sorry, secondary messenger. The primary messenger is the neurotransmitter. The secondary messenger is what the enzyme releases. This enzyme can then open up the ion channel, as you see here, or it can go to the nucleus and other parts of the cell and actually cause other things to occur in the cell. Like I said, it can cause the neuron to change, where maybe it increases neurotransmitter production, maybe it decreases neurotransmitter production. These are all various different things that can occur with this indirect method. So that gets to the exact reason of why is there an indirect method if the direct method is simple and cheaper energy-wise, because this version here, this second indirect method, costs even more energy. It's very energy dependent, or it's very energy heavy, sorry. So why does our bodies do this? Why is there this type of indirect method? It's because it can actually do other things, like this, which you see here with the going to the nucleus, but also because it tends to be a longer lasting effect of opening the ion channel. The one in the outside from the previous slide, the direct method, those get basically broken up really quick, so the ion channel only stays open for a very brief amount of time. However, in this case, with both of these, the ion channel can actually be open longer, so it can cause a greater effect. I hope that makes sense. So, basically what it comes down to, what we've been looking at, is that there's this change in membrane potential that's caused by neurotransmission. Everything we've talked about in the previous set of slides and this set of slides to this point has gotten us to the fact that what is going on is neurotransmitters are binding in certain ways that are changing, not necessarily increasing, not necessarily decreasing. They're changing the membrane potential. So, as I said before, some could increase the membrane potential. Some could decrease it. The point is that these neurotransmitters, this neurotransmission, is just changing membrane potential. And then we have these ligands, like I already said, that are any chemical that binds to a receptor. Why is this important? Why don't we just call them neurotransmitters? Because other things can bind to a receptor other than just neurotransmitters. Neurotransmitter is the standard thing, but things like drugs can bind to them. It can cause increases in things like dopamine because of their binding there. Things like other chemicals and drugs and medications can bind that block neurotransmitters. As I said, when we look at this example here, where we've got on the right, let's say we get something that is circular but has a spike on it. They're not exactly shaped like this, but they're not shaped like this at all. But let's say, just for chance, we get something like that. It is the correct enough shape that it fits in there, but not the correct enough shape that it pushes down on the receptor site to open up the ion channel. The issue there is it then blocks other neurotransmitters from being able to enter that site. And as we already said, the binding sites are locations of receptors where the ligand binds. So this slide is kind of a review of what we've covered to this point. So let's kind of look at what happens then with the neuron itself. So an action potential leads to the release of neurotransmitters from the presynaptic terminal. So you've got the presynaptic terminals on the terminal buds, the presynaptic terminals there, each of the presynaptic terminals, the action potential from the previous neuron, which is the one coming in here, here, and here in this instance. That action potential caused the presynaptic terminal to release these neurotransmitters. It caused those vesicles to come to the surface and burst open. The neurotransmitters then go across the synaptic gap and bind to the postsynaptic terminals, as we see here. So they bind to the postsynaptic terminals either on the synapse of the soma or the synapse of the dendrite. They bind there. Okay, once they've bound there, ion channels linked to the receptors open up, and it's a influx of either positive or negatively charged ions. Let's say it is causing an influx of positively charged ions. Let's say the neurotransmitter that's released binds to channels that open up and allow sodium into the neuron. So there's an influx of these positively charged ions. The postsynaptic neuron will then reach the threshold of excitation, causing an action potential to occur, which then leads us back to the beginning, but now in the next neurons. I hope that makes sense. It's basically a repeating process that occurs. Actually, let's watch a video from Magic School Bus.
The feather triggers the tickle receptors on the neuron. An electrical impulse rockets away from the neuron's dendrites. The message then races along the axon, where the message is delivered to the next neuron. Amazing! A seamless chemical handoff from Keisha's axon terminal to Carlos's dendrites. And there goes Carlos speeding towards the brainstem straightway. Another perfect message transfer. Another perfect message transfer, and now DA is off and racing.
Whoa, talk about speedy delivery!
Yet another perfect transfer.
Next stop, the brain! Here you go, brain!
And the message goes up the brainstem where Jody's brain finds out that...
My nose tickles.
Now she needs to scratch that itch! So she sends a message back to the muscles in her arm. Hand and finger like this, and message received! Job completed! Itch scratch!
Right, it looks like the sound might have been off on that, but you kind of get it. It's a amusing way, a very basic way, but it explains it in a different way than I explained it. Hopefully that worked on that previous slide. Okay, so what we're going to get to now is a couple things relating to what we were just talking about. And that is what's going on with these neurotransmitters and what they're opening up. So you can have various different ion channels opened up, basically causing the different things to occur, like sodium moving into the neuron, causing depolarization, which can cause what's referred to as excitatory postsynaptic potential. So excitatory postsynaptic potential is when you get neural excitation. Basically, this increases the chance of the neuron firing. So it increases the likelihood of an action potential occurring. It causes depolarization. So if you remember from the previous one, depolarization is the start of the neuron firing or the action potential. Or you can have inhibitory postsynaptic potential. This is where the neurotransmitters bind to sites that actually do things like let potassium out. Letting potassium out leads to hyperpolarization, leads to neural inhibition. It decreases the likelihood of an action potential occurring. I do have to point out, A excitatory postsynaptic potential does not mean behavioral excitation. It does not mean you're going to increase a behavior. It just means that neuron is going to fire. Some neurons actually fire to decrease behavior. Some neurons fire to increase behavior. Some neurons fire and have a zero change on actual behavior. So a excitatory postsynaptic potential doesn't mean behavioral excitation. It just means neural excitation. And the same is true for an inhibitory postsynaptic potential. It doesn't mean behavioral inhibition, it just means neural inhibition. Let's kind of look at that in another way, how this excitation and inhibition works. So when you've got excitation, when you've got a lot of neurotransmitters that are causing excitation, it increases the chance of it firing to the point where what you see on the left is you get the neuron ends up firing because it reaches the action potential. However, what happens if you get a little bit of excitation and a little bit of inhibition? The blue is inhibition, the red is excitation. The neuron is typically not going to reach the action potential. It's not going to be triggered because the inhibition is enough to counteract the excitation. So going back to this, did you drop the pot? Did you drop the pot of casserole? You had the neurotransmission from the sensory nerves all the way to the spine. The inner neurons from the spine are telling the motor neurons to activate and drop. However, your conscious thought about not dropping it, about knowing that you're about to touch something that's warm and not dropping it, is sending inhibition down to this red one again, the inner neurons that come from the brain, that are getting messages down from the brain. These inner neurons coming down from the brain are causing inhibition. They're releasing inhibition neurotransmitters, which then causes the muscle, the motor transmitters, to not activate. The action potential doesn't occur in the motor transmitters, the motor neurons, sorry. The action doesn't occur because the action potential didn't occur, so you don't pull away, you hold on to the pot. And long story short, from what we talked about earlier to what we're talking about now, that is how you get to from asking about, do you drop something? How do you counteract a reflex? You counteract a reflex by conscious thought, basically causing inhibitory neurotransmitters to be released. And when we're talking about inhibitory postsynaptic potential, neurotransmitters that are causing inhibitory postsynaptic potentials. Let's talk about a special case. I told you we'd come back to this, and that's axoaxonic synapses. Axoaxonic synapses, why are we talking about them now? Because they actually act in a way like those inhibitory or excitatory postsynaptic neuro things. Postsynaptic, sorry, my mind just doesn't want to work. Postsynaptic potentials. They cause postsynaptic potentials that are inhibitory or excitatory in such a way that they actually, what they do is they modulate the presynaptic terminal of the terminal bud that they are touching. So yes, you've got a postsynaptic here. The same terminal bud here has both presynaptic and postsynaptic. It's got postsynaptic to the axoaxonic terminal or the axoaxonic synapse, meaning you've got a terminal touching the side or the top of this terminal. But what it's going to actually do is it's going to create presynaptic modulation. It's either going to increase the amount of these vesicles that reach the surface and the amount of transmitters that are released, or it's going to decrease the amount of those vesicles that reach the surface and the number of neurotransmitters that are released. So it basically is another, and quite often this is a terminal bud on the same neuron. So it's not a terminal bud on a different neuron, it's a terminal bud on the same neuron. Sometimes there'll be terminal buds on different neurons, but often these are terminal buds on the same neuron, and they're basically acting as a form of modulation. The last main thing we're going to talk about here is basically how the neurotransmitters are stopped, and how the synaptic gap is turned off in a sense. So one of the things that occurs is what's called reuptake. Reuptake is the most common, and this is where neurotransmitters are taken back into the presynaptic terminal. So the presynaptic, you remember, that's the one that released the neurotransmitters. The presynaptic terminal sucks these neurotransmitters back up, and it actually does it pretty quick. These neurotransmitters are released, and then they're sucked back up really, really quickly. It's a constant cycle that's going on where neurotransmitters are being released, sucked in, released, sucked back up in, released, sucked back up in. This is actually the most common type of thing that happens, is these neurotransmitters are sucked back into the presynaptic terminal. What's going on here is once enough neurotransmitters have basically activated the next one, other neurotransmitters are doing nothing there. So by sucking them back up in, it clears it and it readies for it to fire again. Plus, it's getting enough back for if it has to fire again, there's more there. And this is a constantly recurring process. Reuptake, release, depending on if the action potential occurs. Another thing that occurs is enzyme or enzymic deactivation. Basically, metabolism is working. The neurotransmitters are restructured by enzymes, broken down to the point where they're no longer neurotransmitters, where there's no longer postsynaptic locations for them to bind to. And then finally, you've got a neural off switch, which is basically where autoreceptors cause the neurotransmitter release to be inhibited. So even if the action potential is released, sometimes what you've got going on due to basically different things that are going on throughout the body is that the neurotransmitter release can be inhibited even if the action potential is being released. This isn't something I'm going to test on. This is just something to kind of finish up the set of slides looking at. So let's talk about these steps in neurotransmission. This is what we've looked at all chapter. You've got chemical communication from one neuron to another. You've got synthesis, which is the neurotransmitter is made by the cell body. You've got storage. The neurotransmitter is stored and protected in the neuron. Then you've got transport. The neurotransmitter is transmitted by the tubules from the cell body to the axon terminal. From the cell body down the axon to the axon terminal through the microtubules. Then you get release. The neurotransmitter is released from the presynaptic terminal. So the vesicles that are holding the neurotransmitter reach the surface, they're released, and then they're released when the action potential occurs in the neuron they're in. So the action potential occurs, this causes the vesicles to go to the surface and release the neurotransmitter. Then activation and binding occurs, where the neurotransmitters bind and activate in the postsynaptic terminal, then causing that other neuron to either fire or be inhibited, depending on what is there. Typically, it's going to cause that neuron to be fired as well. And finally, deactivation occurs, where neurotransmission is stopped through reuptake and metabolism. Seems like a very complicated process, but this is a complicated process that is happening essentially millions upon millions of times in your body, repeatedly, over and over and over again. This is a very repeated process happening all the time. When you are thinking, when you are talking, when you are moving, Even when you're sleeping, this process is continually occurring in the brain and in the body. So in conclusion, we talked about communication between neurons. We talked about synapses and the synaptic gap or the synaptic cleft. We talked about neurotransmitters. We'll talk more about those later. We talked about postsynaptic potentials and excitation and inhibition, and we talked about deactivation. I said this was a relatively short chapter or relatively short lecture here. It's just finishing it up from before. I could have mixed them together into basically an hour and a half, hour and 45-minute lecture, but it's just better to break this up into two just because there's just so much information. Next week is going to be very similar to lectures that are a lot of information and it's broken down into halves. Thanks, come on back.